Welcome to the 2019 Robert S. Gordon Jr. Lecture in Epidemiology. My name is Stephanie George. I'm an epidemiologist in the Office of Disease Prevention, and I'm pleased to represent our director, Dr. David Murray, today and introduce our speaker. Before I do that, I wanted to invite everyone to join us after the lecture for a reception in the NIH library directly to my left. The Robert S. Gordon Jr. Lecture is awarded each year to a scientist who has made major contributions in the area of research or training in the field of epidemiology or the conduct of clinical trials. The award was established in tribute to Robert S. Gordon Jr. for his dedication and contributions to the field of epidemiology and his distinguished over 30-year service to NIH, during which time he served in numerous senior leadership positions. Now let me introduce our speaker, Dr. John Ioannidis. John comes to us from Stanford University, where he is the C.F. Remberg Chair in Disease Prevention, Professor of Medicine, of Health Research Policy, and by courtesy, of Biomedical Data Science and of Statistics. He's also the Director of the Meta Research Innovation Center and the Director of the PhD program in epidemiology and clinical research. Dr. Ioannidis received his MD from the National University of Athens in 1990 and also received a doctorate in science in biopathology from the same institution. He trained at Harvard and Tufts in internal medicine and infectious diseases. His awards are numerous, as are the disciplines his work spans. I encourage you to check out his biography online, and I will now, with no more minutes, introduce our speaker. Dr. Ioannidis' presentation today is titled, "In Scientific Method, We Don't Just Trust, or Why Replication Has More Value Than Discovery." Please join me in welcoming our 2019 Robert S. Gordon Jr. Lecture Award recipient, Dr. John Ioannidis.
Thank you for this tremendous honor and for asking me to give this lecture today on this topic. It's always a pleasure to be back at NIH and to meet with colleagues and to meet more colleagues, very bright people. This is a stellar institution, very unique in the world. So in scientific method, we don't just trust. Trust is great to have, but we need to have more than just trust. Science is a fantastic enterprise, very successful, about 180 million papers floating around. This is just 20 million of them from the last 16 years. They create a universe along with 2 million patents and 200,000 disciplines of science. I'm pointing with an arrow here, and it's very tiny font, but let me magnify that. Hi there, my best paper is a speck of dust and a speck of dust and a speck of dust somewhere around here. No single paper can ever compete with its surrounding or with science at large. Science is a communal effort. It's that whole galaxy that matters. It's a living galaxy, an evolving galaxy, with plenty of data, with plenty of hypotheses, with plenty of inferences. At the same time, that galaxy also has plenty of empty space, plenty of dark matter. And what would that dark matter be? It would be analyses that have never been published, that have been done but are not published. It would be data that are available but they're not really available. They were accessed maybe once by someone, but then they were lost. It would also be science that was not done. It would be replications that were not done because it was thought to be Me Too research, and also lost opportunities about research that never flourished because it was not funded, because there were not enough resources to do it. So how can we look at the existing matter and try to understand how we can expand that universe further? Much of the emphasis has been on making discoveries. But to be honest, I think that discovery is a boring nuisance. Almost every single paper that you will read in the literature will claim that it has some novel results. Every grant that I will submit to NIH I promise to find something novel, and I know that probably I'm lying, statistically speaking. This is a text mining exercise probing the entire PubMed from 1990 to 2015. 96% of the biomedical literature claims significant, statistically significant, and mostly novel results whenever p-values are used, either in the abstract of the paper or in the full text papers that are available in Medline. Translation proceeds at a glacial pace. Despite having all these millions of statistically significant and seemingly novel results, we do make progress, but it's not millions of discoveries that move to have that tremendous impact on medical care, on patient outcomes, on health, on living better and longer lives. Several years ago, we looked at the creme de la creme, the most highly cited clinical research ever published. These are shown here with red landmarks, milestones, and we asked how long did it take to get there, to have one of the most highly cited papers in the history of medicine. On average, it took about 25 to 30 years. In the case of nitric oxide, it took about 200 plus years. There are a few exceptions where things materialize faster. For example, I consider one of the most exciting experiences in my career when early on, as a scientist, I was at NIH. I was involved in ACTG 320, a randomized trial that showed that with triple therapy, we could dramatically decrease the risk of death and disease progression for HIV-infected patients. That trial was published within less than 4 years from the time that protease inhibitors had been developed based on a concentrated and communal effort to try to do something about HIV. And it did work. So how can we shift from stories where it takes 200 years or 30 years to stories where it takes 4 years? And also, how can we shift not only the timing, but also the credibility of these efforts? Because in that slide here, I also have some black milestones, which is when larger beta-controlled studies were done that showed that that creme de la creme paper was actually either completely wrong or exaggerated in terms of what it was promising to have found. I think that we are incentivizing a fake narrative. The narrative that is dominant is that we have an oversupply of major true discoveries, and I think that this is currently untenable. I don't need to remind you how few new substances, new drugs get licensed, and even though we're trying to push that agenda, and we have the brightest minds in the world working on science, the effort that we put is tremendous, and the real progress that you can measure in any way you want is far more limited. Now, this narrative coexists with some additional urges. One of them is make haste, rush. Patients are dying. License new drugs immediately. And I do feel that pressure, and I think that, yes, we need to make haste, but this doesn't mean that we should cut corners in terms of the evidence that we need to accrue. Second urge is to be methodologically sloppy. Oh, just get an answer right away. We have all these huge, routinely collected data sets. No need to do randomized controlled trials. Just run something through the mill of your computer, and that's going to be accurate. And very often, this is not accurate. And the third urge is avoid replication. Don't waste time with replication. This is Me Too effort. We need to move forth to even more discoveries. Now, the value of discovery can be modeled. So here's a very simple model. Let's say that R is the pre-study odds of the research finding being true. BF is the base factor that is conferred by the discovery data. H is the ratio of the weight of negative consequences from a false positive discovery claim versus the positive consequences from a true positive discovery. Then the value of the discovery process is proportional to true positive minus H times false positive, or if you work through that, it is proportional to R times the base factor minus h. And obviously, you can add a constant. If you have 2 scientists, 2,000 scientists, 2 million scientists, you can multiply that with how much effort is going into this. R and H are rather field-specific. If I have wasted my life in a field where there's nothing to discover, that's very sad, but that field just has nothing really useful to discover. If I want to make progress, I just need to change fields. Sometimes, maybe there are things to be discovered, but they would require a completely disruptive approach, abandoning what we do and just taking a completely new approach to trying to attack the same question. H is also pretty much field-specific. So the focus should be, must be on increasing the base factor of the work that we do, the information content of the work that we do. The options for increasing the base factor are numerous, and they depend on what field we're working with, but typically running larger studies, so getting more evidence would help, and also ensuring greater protection from biases would help, which means that we optimize our design, our statistics, our thinking ahead of time about how we should go about answering a question. In that very same equation, it's very easy to get negative values. If you want to avoid having negative values from discovery, then one needs a base factor to be more than the ratio of H over R. And often this is very difficult. Why is that? Most original discoveries are claimed to come from small studies where biases are very common, and therefore the base factor often is even less than 5, which in the Bayesian work means very, very little. And also most fields currently are working in areas where the pre-study odds are pretty low. We're attacking massive spaces of exploration where there are signals to be discovered, but the denominator of all the potential things that we can measure and all the things that we can analyze is probably very large. If you have that combination, most discoveries are operating in a space where they have negative scientific value. It's far more likely that they will confuse us, that they will generate false negatives that would lead people astray and more resources wasted downstream building on something that is a false negative claim or an exaggerated claim, rather than that they will save the world. How do we sort out where to go next? We need replication. We need to take all of these tentative discoveries and try to replicate and see what still survives different efforts to reproduce these results either exactly the same way or with different angles of triangulation. Is that new? Not necessarily. This is pretty much how science started in the Western world. You had to replicate and reproduce findings in front of the Royal Academy, and people were watching to see whether the apparatus would work. Currently, we mostly trust that what was done behind closed doors or behind a closed computer is something that we can put trust on. We do have empirical studies on fields where replication is not just considered to be a me too type of effort, and actually replication practices are common. And these empirical studies suggest that most of the initially claimed statistically significant effects are either false positives or substantially exaggerated. One such field is genetics. Genetic epidemiology went through an immense transformation over the last 10 years, moving from candidate gene studies, where people had to come up with single hypotheses to test in an agnostic fashion with much larger sample sizes and consortia, the whole genome, more or less, in terms of association with phenotypes. By doing this, one could go back and assess how often the papers that were published in the candidate gene era were successfully replicated through genome-wide association approaches. The replication rate, on average, was 1.2%. This may be an underestimate because of power considerations. I'm willing to take that to 5% or maybe 10% at most. but more than 90% of these papers that were published for many, many years, or over 20 years, in the very best of our journals were probably not saying much and probably just false positives. Here's another evaluation, animal studies. There's tens of thousands of animal studies being done, and I think that they're tremendously important because animal research is an essential, indispensable gateway to human clinical research. I mean, if we decide to just move directly from very early bench discovery to humans, I think we will be testing lots of noise inadvertently on humans, and we definitely don't want to do that. However, you're all aware that for many fields where we have hundreds, if not thousands, of potential leads from animal studies, like neurological diseases, we hardly have any successes when it comes to humans. For example, dementia or treatment of stroke. And we have a couple of treatments for stroke, but we have hundreds of treatments that seem to work in animal models and their level. One might say that the explanation for that is that animals are very different from humans. And I think that there are differences. I hope I'm a little bit different than a mouse, but my genome is not that different. And I think that most of the problem is not necessarily the dissimilarity of the experimental system as the way that research is done in ways that bias can creep in. This is a slide from a paper where we looked across the entire literature that we could get on animal studies and neurological diseases, and we found that very prominent signals of excess significance bias, pressure to deliver statistically significant results could be detected across that field. And once you started sorting out different hints or patterns of bias, there were very, very few pieces of evidence that remained to be pretty strong. Preclinical research. In the last 8 years, we have seen rapid change in our understanding about replication and reproducibility. This time, most of the leads came from the industry, and the industry had every right to feel uncertain about not being able to reproduce papers published in top journals by academic teams. For academic investigators, maybe this is curiosity, maybe this is a paper in Nature, maybe it is a way to get tenure. For the industry, it was an issue of spending $500 million on something that would lead nowhere. So we had several companies that launched reproducibility checks on high-profile, highly cited papers coming from academic teams, pretty much summarizing what they had already been seeing in large scale. Most of the results could not be reproduced in their hands. The reproducibility rates range from 0 to 25%. In one such famous example where Amgen could only replicate 6 out of 53 landmark studies for oncology drug target projects, Glenn Begley concluded that the failure to win the war on cancer has been blamed on many factors, but recently a new culprit has emerged. Too many basic scientific discoveries are wrong. We have seen similar reproducibility checks across very different scientific disciplines. Psychological science has also gone through a major transformation. This is a paper that was published 3 years ago in Science, a collaboration of 273 psychologists and their teams trying to reproduce 100 of the creme de la creme papers from their top journals. And you can read these results in different ways, but the summary, no matter how you look at it, is that about 2/3 of the time, the original result could not be reproduced. And obviously, the effect sizes were much smaller compared to the original. What if you only read Nature and Science? This is another reproducibility check of 21 papers. On average, the reproducibility effort revealed an effect size of 50% of the original. And in many cases, there was nothing there. The effect was completely on the vicinity of the null with pretty tight confidence intervals. Does it mean that the reproducibility was correct and the original was wrong? I mean, all of these cases are efforts where people systematically try to follow the exact recipe as the original study. And they even communicated with the investigators. They even tried to make sure that they have all the details that would be critical. So there could be many explanations. It could be that both are correct, but for somehow we don't know some reason why they disagree. It could be that both are wrong, because again, there's some biases that are creeping in that we're not familiar with. Or it could be that one of them is not correct. But clearly, if you have a situation where under the very best efforts to reproduce, you cannot get something to work again, one has to wonder, would it work if we were to use that widely in real life, in patients, in communities, in the real world? Reproducibility efforts can be tricky, and they can also be emotional. They can lead to what I call the reproducibility wars. The reproducibility effort, for example, on cancer biology, started publishing a number of papers where that very meticulous effort to reproduce high-profile cancer biology papers were attempted to be reproduced. Everything had been pre-specified. There were pre-registered reports. The results were very clear, but then most of the time, if the original was not reproduced, the original investigators would fight back. And you end up in a situation where you feel that reputation is at stake. There's very fierce emotions about who is right, who is wrong. Careers are thought to be at stake, and interpretations can be different on what is successful and what is unsuccessful. This means that people do care about reproducibility, and they should care. It's really a central piece of the scientific method. However, what exactly do we mean by reproducibility? What is research reproducibility? If you look across all 22 major disciplines of science, there's a rapidly increasing use of that terminology if you do a text mining exercise like what I'm showing you here. But people mean very different things. Basically, you can separate reproducibility into three main clusters. One is reproducibility of methods, which is the ability to understand or to repeat as exactly as possible the experimental and computational procedures, so availability of software, of script, of data, and the ability to put them together and get the same result from the very same data. Reproducibility of results, which means that we're doing yet another study on new participants, new samples, new observations, and we hope to get a result that is consistent, compatible, ideally as close as the original. And reproducibility of inferences, which means that we have one study, a replication, or multiple studies, or a body of evidence, and I ask people in the audience, what do you conclude out of this? And we may or may not agree about what these data and what these results mean. Reproducibility can be affected by the recipe of research practices that we apply. And you can think of two extremes of research practices, two stereotypes. Of course, this is a stereotype. It doesn't mean that I have some particular researcher in mind who's doing things wrong. But if you think of small data and big data, this is what often that looks like. So with small data, which is still the majority in most scientific fields, we have the prototype of the solo silent investigator with a small team, very limited resources, working in competitive environments where there's limited funding. Unavoidably, the studies that will be done will be pretty small. One needs to be successful. You know, these 3 or 4 years of funding end, and you need to say, I have something major to say and something major that would lead me to my next grant. Actually, you need to get going, probably not after 3 years, but after 3 days to write the next grant. And how do you do that? Much of the time, the results will be quote-unquote negative or unimpressive. You need to start exploring, searching whatever space you have available. There will be a lot of cherry picking of the best looking results, a lot of post-hoc interpretations. P-values of just slightly less than 0.05 are typically considered to be enough. And with p-hacking, it's almost always possible to get there. There's no registration because that decreases the degrees of freedom. No data sharing because it offers ammunition to competitors. And no replication because if you try replication and it fails, you have invested twice the effort and you're back to square zero, and people will say you found nothing, so that's it for you. Some of the ways to improve the validity of small sample size research are very easy to implement. They cost nothing. They would save us from a lot of trouble. However, they're not implemented. For example, we know about experimenter bias for over half a century now. Rosenthal published his papers more than half a century ago. And we know that in animal experiments or in other in vivo studies, but also in vitro, it's great to have people reading the results to be blinded to the experimental conditions. However, that happens less than 10% of the time. Randomization, we know that it's not about humans here, it's about animal work or other experiments. It costs nothing, it's very easy to do. It would save us from lots of trouble of imbalances that would be consciously, subconsciously, or unconsciously be created between the compared groups. Again, less than 30% of the time, even in the most recent studies, this is implemented. With big data, we're challenging the status quo of small study research, but it doesn't mean necessarily that our odds of success are necessarily better. In that situation, which is becoming more common with electronic health records, with large omics databases, and other such efforts, we have extremely large sample sizes. We have overpowered studies that are likely to give signals no matter what. Even if no signals are worthwhile detecting, you will detect tons of them. This means that, again, there needs to be a cherry-picking process. And again, a lot of that is done post-hoc. Actually, most of these databases are not even assembled for research purposes. They just happen to accrue overnight. You know, I'm sleeping, and I wake up in the morning, and there's so many more patients that have been added to the electronic health records that one or more researchers could tap into. Statistical inference tools are a bit different. People recognize very quickly that if they just use a p-value of less than 0.05, everything will be statistically significant. So there's a little bit of more sophistication much of the time in that space. But very often you see idiosyncratic statistical inference tools without much consensus. People may be working in the same or very similar fields, but nevertheless, they're using very different approaches and very different thresholds for claiming success. Registration remains an exception, also in that space, and data sharing does happen probably more often than in fields that use small data sets, but unfortunately, much of the time, there's no understanding about what exactly is being shared. Most of the time, these data sets are just dumped somewhere with some access where someone could easily or more difficult get access to them, and then you ask, does anyone know what these data are and what they show? And literally, even the investigator who has generated the data probably does not know, because they're just a big black box that is very, very hard to interpret what exactly has been generated, let alone how valid it might be. Replication does happen in some fields, and I showed you some examples. And even when it does not happen, we may have a situation where people say, I'm doing a study that is somehow different. Actually, there's an incentive to justify that I'm doing a different study, but then you look at the two studies and say, well, you're asking exactly the same question. But to get it published, someone has to say it is different. Meta-analysis can take these similar studies and examine them in their totality. Studies on the same question with pretty much the same comparisons, with pretty much the same interventions or the same risk factors or the same hypotheses being addressed, and give us a sense of heterogeneity between these different studies that address fairly similar questions. Heterogeneity may be genuine. Much of heterogeneity may reflect genuine differences across these studies. However, it can also point out to differences that stem from biases that are differentially expressed or affect differentially different investigations on the same question. If that's the case, you should be able to pick some hint of the presence of a bias because it leaves a pattern of a particular type of diversity, a particular type of heterogeneity across the results that are being combined in a meta-analysis. So this is pretty much what we did here. We pre-specified 17 patterns of bias, and we thought, how would the literature look like if these biases were present? And for all the meta-analysis that we could find across all scientific fields, when there were many, we just selected a random sample thereof, we tried to ask, do we see these patterns? Most of these biases could be seen in most fields. However, some biases seem to be having more common hints of their presence in some fields compared to others. For example, small study effects, which is a pattern where small studies give more prominent, perhaps exaggerated results compared to larger studies, was a pattern that was seen very prominently in the social sciences, was seen prominently in the biomedical sciences, and was not really seen, or very soft signals were seen in the physical sciences. Physical sciences are much better used to working with large data sets, with communal science, with CERN or big telescopes, sharing all the information across all the participating physicists or astrophysicists who are working on a particular domain of science. The same applied to other disciplines. Sometimes one or another field had more of a signal of one bias or another, but most biases were seen in most scientific fields. What are some potential solutions? There are many solutions that are being proposed. They're integral to the process that we do science, and you can think of the reproducibility problems as a central concern in everyday experimentation, in everyday study design, in everyday ways of running science. This means that lots of people are thinking about them, and some very smart solutions have appeared, but also many of them are very speculative. They have no empirical support to tell us that they will make things better rather than make things worse because of collateral damages that they may procure. Here's 12 families of solutions. Large-scale collaboration, adoption of replication culture as a sine qua non, registration, sharing of data, reproducibility practices and checks, containment of conflicts in sponsors and authors and other people involved, more appropriate statistical methods, standardizations of definitions and analysis whenever this is possible, more stringent thresholds for claiming discoveries or successes, improvement of study design standards, improvement in the peer review, reporting, and dissemination of research, and finally, better training of the entire scientific workforce in methods, in the scientific method and how it should be applied, and statistical literacy and numeracy. I hope I have convinced you by now that some of the most successful fields in terms of credibility are those that have adopted large-scale collaboration and adoption of replication culture. This is one example of Manhattan plots. Genetic epidemiology was transformed from a field that almost nothing could be replicated to a situation where we can pretty much safely reproduce signals. I'm not delving on whether these signals are useful, on whether that's something that you can take to patients and really change their lives, but at least they're there. And sometimes the signals may be true, but maybe they're not going to be useful. This is perfectly fine. We know what we're dealing with. We know what is the true complexity of the research questions that we're facing. Registration can be tricky in many situations. The least that I would like to see is people who are doing very interesting exploratory research feel that they need to say that I have registered my study. If you wake up at 3 o'clock after midnight with a fancy idea that you cannot go back to sleep and you need to run to the lab to try it out, and you're just messing right and left with different possibilities, probably you didn't write a protocol before you started doing that. But something very interesting may come out of it, and maybe you're the next Alexander Fleming with penicillin. How likely is that to be the case? Not very likely, but there are 20 million scientists authoring scientific papers. So a few among those 20 million will be Alexander Fleming, hopefully. What we need to do in that case is just say that that was exploration. It was mad, wild, crazy, exciting, fascinating exploration. That's what came out of it. Now someone needs to reproduce it in a prospective reproducibility effort. Level 1 would be registration of a data set. I feel very uneasy when I'm working in a field that I don't know how many data sets are out there that could be probed and in how many different ways they can be probed. Registering a data set is like registering a nuclear arsenal. So I'm telling you that I have this big data set in my computer. It includes observations on 2 million people, and I have 2,000 variables on each one of them, which means that tonight, if I feel depressed, I can press a button on some statistical software and launch so many billions or trillions of chi-square p-values against you. So it's one way to confer the breadth of the possibilities of analysis that can be done. Level 2 would be registration of a protocol, if a protocol exists. There's lots of research where there's no protocol, and sometimes it's just blue sky exploration, but most of the time a protocol is feasible and should be there before something is done. Time spent on coming up with a protocol, I think, is always well spent. Level 3 would be registration of the analysis plan, if there is an analysis plan. Sometimes you have to acknowledge that this is the analysis plan that I could think ahead of time, and these are some modifications that arose because of some peculiarities that I had not anticipated. Level 4 would be registration of both the analysis plan and the raw data, and level 5 would be open live streaming, where you iteratively communicate with the rest of the scientific community about what are the experiments that I'm planning to do. One receives feedback, they're revised, they're done, they're shared again, and this is an iterative open process with the whole community. This is pretty much how the claim by NASA that they had identified bacteria using arsenic instead of phosphorus for their DNA backbone was refuted. It was a paper in Science. In the absence of having some rules in the science game, in many fields, any result can be obtained. What you get is what I call extreme vibration of effects, and at the extreme you get the Ioannidis phenomenon, being a Greek-Roman god who could see in two opposite directions. These are some data from the National Household Survey, extremely meticulously collected information, and if you focus on the right panel, this is hazard ratio on the horizontal axis minus log 10 p-value on the vertical axis. This is the association of alpha tocopherol, or vitamin E levels, with the risk of death. And there's 1 million points on that plot. There's 1 million different results that one can get in the very same data set on the very same question, just analyzing the data slightly different. For example, death can be affected by zillions of other factors. So if you account for a factor or not, you have two choices. If you have 19 such choices to make, this is 2 to the 19th power, this is 1 million different possibilities of analyzing the very same data on the same data set on the same question. 70% of the time, vitamin E decreases the risk of death. 30% of the time, vitamin E increases the risk of death. If I have a strong belief on what vitamin E should do, I can get that result, no matter what. This is really happening on a daily basis. It is happening on some of the most influential papers that you will see in the literature. This is a paper that last year was one of the 20 highest impact papers across all science, and practically it concluded that with 3 cups of coffee per day, your risk of death decreases by 17%. If these wonderful colleagues could give me their data set, I can make it be that your risk of death will increase by 17%. It's an open pledge. Transparency. How can we find out that we can even trust the data? That there's not an A missing from transparency, for example. We need to find ways to make sharing easier. This is a pivotal study Study 329. It sounds like a submarine, U329, but actually it's a randomized trial. And when it was done and published in 2001 by Smith, Klein, and Beecham, it showed that paroxetine and imipramine are very effective and very safe for major depression in adolescents. 15 years later, the very same data set was reanalyzed by independent, non-industry affiliated investigators, and they concluded that paroxetine and imipramine are not effective and they're not safe for major depression in adolescents. How often does that happen? A few years ago, we looked at all the reanalysis that had been done on the same clinical question from the same clinical trial data set. These are reanalyses rather than replications, but if we cannot reanalyze and have some confidence, why should we even take the second step of doing a separate independent replication on a new study? We found 37 such reanalyses, and 35% of the time, the conclusion, the main conclusion of the reanalysis was different compared to the conclusion of the original paper. This treatment should be used? No, this treatment should not be used. It should be used in this subgroup? No, it should be used in that subgroup. Was it research parasites who had run these reanalyses? Was it rogue analysts who were trying to make a career of themselves or put, the great original investigators into shame? Almost always, it was the same original investigators who published the reanalysis. But they published it in an environment, in a publication environment, where if they were to say that I tried to reanalyze my data and I found exactly the same thing and I conclude exactly the same conclusion, someone will tell them, why did you waste your time? This is duplicate publication. So our incentive system selects for confusion, selects for discordant results, even for things that are done by the same investigators. Recently, we revisited that pattern, looking at another extreme where reanalysis actually should have been the default. PLOS Medicine and BMJ have policies in place that if you publish a randomized trial, you need to make not only the full protocol, but also all the raw data available to anyone who would ask for them. So along with Florian Odette and other colleagues from my metrics team, we invited investigators who had published under this policy to share the data with us, and we promised that we would reanalyze them at no cost for free. It's a little bit like getting an invitation from IRS, but I'm really glad that almost 50% of these investigators actually did send us their data, and they also were very helpful with that, and we reanalyzed the data. We found a few errors, but nothing major. The conclusions will still remain the same. So you have two extremes. One is a highly selective environment where people are just trying to impress and share very little, and the other, a very selective also environment where everyone is willing to share, or at least 50% are willing to share, and they're very open to having their data reanalyzed, and then everybody looks fine. One might argue that maybe these people who did contribute their data took an extra look, and if they found any major errors, they made sure that they send us a version that would be compatible. I don't want to become paranoid. I believe that if we create a culture of sharing, most of the results that we will get, if people are well trained, hopefully will be reanalyzable and reproducible at that level at least. So how do we improve sharing? Sharing is a challenge in itself, and when we tried to retrieve the most highly cited papers, data sets, the raw data behind the most cited papers in psychology and psychiatry, we met with quite a lot of resistance. These trialists and these studies were not bound by the PLOS Medicine and BMJ rules, so they could or could not share or might not share their data with us. We realized that in some cases they had made their data available already, and in a few more they were willing to share that information, but very often they could not or did not. What were the reasons for that? The most common reason was that it was outside of the researcher's control. I have come across many, many situations where researchers do not control their own data. Sometimes researchers publish as first or last authors or both, and they have never seen the data that they put their names on. It's really scary. Legal and ethical concerns. The consent did not allow that, and that was done in the 1990s or so. Preparing our own sharing system. The data no longer exists. The classical example in the literature, the data has been eaten by termites. It's a famous quote. Insufficient resources, or researchers are still using data, and the question is for how long? For 6 months, for 2 years, for 20 years. Are we making any progress in sharing? We are. A few years ago, along with Muin Khoury and Shelley Scully from NIH, we looked at the reproducibility research practices and transparency practices across the biomedical literature, and we found that hardly anything was being shared between 2000 and 2014. There are some niches like genomics that are doing this, but in the big view of zooming out, that was very, very uncommon. Conversely, when we looked at 2015 to 2017, there was real progress. And in some cases, that progress can be explained because journals did change their policies. For example, in psychology, with that revolution of reproducibility, some journals switched to encouraging routine sharing with a badge system, and you see a rapidly increasing rate of sharing data sets in the published papers in these journals. But in biomedicine, I think it has been more of a diverse movement where multiple journals, multiple fields, multiple investigators, multiple institutions are incentivizing or facilitating sharing. So the rate has gone from literally close to 0% to something like 25% by 2017. We also see some other concomitant changes in transparency indicators. For example, disclosure of funding or disclosure of conflicts of interest has gone up over the years. Most people still claim that they have something novel to say, even if you look at the abstract, it's very prominent. But there are more people who say that I'm trying to replicate something, or at least trying to do something novel, but at the same time replicate existing knowledge. Computational methods can facilitate much of our reproducibility quests. A couple of years ago, we published these guidelines trying to enhance ways that journals, investigators, and institutions could move forward in improving their reproducibility for computational methods, including software, script, and linked to data. Very often you see a link and you click on that link and there's nothing there. You get an error signal. So there are some very easy interventions that can improve the availability of these functional links, but there's also some more sophisticated ones that can take us substantially further. Better statistics and methods. We have seen a transformation of research over the last several decades, and data science has taken a central role across multiple scientific fields. Is that new? Well, science has always been about data, but I think that while in the past it might have been easy to work with small data sets for descriptive purposes, currently you really need a license to kill, a license to analyze. And most of the time, most of the papers that I see probably are done by investigators who don't have that license to analyze. It's very uncommon to see transparent statistical analysis plans. We repeatedly lament that we don't invest much in training our investigators in statistics and issues of design, and that good study designs are underutilized. I mentioned the very simple choices of randomization and blinding of investigators that are so underused. How do we do that? Do we just use some checklists? Do we add another level of bureaucracy, for example, and we say, well, fill in that checklist? Is that enough? If someone has done something in a way that is very substandard, if it's really silly, how likely is it that it will be acknowledged rather than just someone have checked that checklist? Oh yeah, I did that, I'm okay. It's a continuous tension. Are we using the right statistical methods? There are many pleas to try to change the way that we make statistical inferences, and I'm not going to spend much time on them because each one of them has a different philosophy in mind. So one such plea is to become more stringent. Many fields that have improved their track record did become more stringent. Genetic epidemiology moved from p-values of 0.05 to a genome-wide significance level of 10 to the minus 9, and things seem to be working much better in terms of reproducibility. For many fields that are working with p-values of 0.05, a very simple move would be to shift to 0.005 for statistical significance, and that would immediately eliminate probably a very large segment of noise, but that would also take away some genuine signal. So this needs to be balanced in each field in terms of whether it's a good idea or not. Most fields are still using null-hypothesis significance testing, but probably this is not a good choice for many applications, including, for example, developing a prognostic score or assessing a diagnostic test or evaluating a therapy or mining electronic health records or mining big data. We need to find statistical methods that are fit for purpose. And very often these may be Bayesian, these may be false discovery rate based, sometimes they may well be frequentist. Who's going to do that? We don't really invest on training our investigators and retraining them with continuous education on statistical methods and design issues. We just try to catch up with the next tool, next technical tool that may be available, but not the core of the scientific method. Conflicts of interest. I think that there is improvement in transparency of reporting conflicts of interest, but very often I wonder, is transparency enough? Can we leave the generation of original knowledge and replication to conflicted stakeholders? Who are the stakeholders who should be running sensitive studies like randomized trials, meta-analysis, cost-effectiveness analysis, guidelines? NIH has been shifting away from most of those. And there's some empirical evidence that if you look at large randomized trials supported by NIH, before registration, a good proportion of them were statistically significant in their results. After registration, this happens very, very uncommonly. Should public entities like NIH resume and expand their role about supporting sensitive research where conflicts really need to be avoided thoroughly if we want to have full trust on them? Should we wait for perfection? No. Science will never be perfect. It's the best thing that has happened to human beings, to Homo sapiens sapiens, but it's a process in evolution. We need to use the best science that we have. So very often I hear, oh, okay, let's get rid of all that junk, that horrible research, and we know nothing. That's not true. We have lights, we have this wonderful amphitheater, the projector is working, I can move my slides. Lots of things have happened, and I think that we need to defend science, and there will be many anti-science voices that are trying to dismantle the scientific effort. There are two ways that we can go about it. One is to say that science is perfect, and probably that's not going to leave us very much room to play, because very quickly we will meet with the scientific method itself, which says that science is falsifiable, and very often we may be wrong, or that science is our best shot. And I think that this is what we need to defend. Sometimes our best shot probably will have lesser credibility compared to other times. We know with high certainty about climate change. We know with high certainty that tobacco is going to kill people. We know far less on whether broccoli is going to make me live longer. So we need to be transparent about what we know and what we do not know. We need more research on research. And I'm clearly biased here because this is where I am investing my effort. So probably it's like I'm asking for more funding. But I think that we need to study how exactly to evaluate our research practices. We need to find ways that you can refute everything that I told you today with more empirical data and with better science. We need to find how we can best perform research, communicate research, verify research, evaluate research, and reward research. What scientific workforce and what world of science are we envisioning? We can model that. We can model science in 2030 or 2040. Is it going to be accurate? Well, I think that we may well be wrong. But this is one such model of science in the future. We used 11 equations to try to describe a simplified universe of science where you have three types of scientists. You have the diligent scientists, who are the majority. You have the careless cohort of scientists who might be cutting a few corners, or maybe they're not very well trained, and they may be following some suboptimal research practices. Now, how many are they? There are many surveys about that. When you ask people, are you cutting corners? The answer is usually no. When you ask people, do you know other scientists in your environment who are cutting corners? The answer is almost always yes. But I think that, let's say, these are the minority. And then you have the unethical cohort, which are clear fraud, creating data that don't exist. So fraud is very, very uncommon. We're talking about less than 1%. If you incentivize these cohorts with the same incentives, if you discover something, if you make a claim, you publish a paper in Nature, you will be fine. And you don't have differential incentives based on their reproducibility, what you will get is that the unethical and the careless cohort will take over. The reason for that is that they can get there faster with fewer resources with cutting corners. So we need to re-engineer the reward system, and I will leave you with a couple of slides on how we do that. We try to incentivize productivity, and productivity is wonderful, but we need to think about the whole electrocardiogram. We need to think of PQRST, which is also quality, reproducibility, sharing, and translational impact. We need to find opportunities to change the way that we do science in our everyday environment. I think it needs to be a grassroots movement. We cannot just wait for some king of science to impose with his or her authority, or queen of science, what exactly should be done. I think it's scientists who realize what makes their scientific work more credible, more reproducible, more applicable. We also need to convince other stakeholders, and they may have other priorities. As scientists want to publish a lot. They also want funding for their work. But there's also the industry, there's private investors, public funders, not-for-profit funders, editors, publishers, societies, universities, research institutions, non-scientific staff, hospitals, insurance companies, governments, federal authorities, people. Some of them want to see papers, others want to see funding, some want to see things that work, and others want to make profit. It's all fine, but it needs to be integrated to try to get the best possible science. To conclude, I think that the presumed dominance of original discovery over replication is an anomaly. It may happen occasionally, but it's really the exception. Original discovery claims typically have small or even negative value, and science becomes worthy mostly because of replication. The reproducibility and the usefulness of many disciplines of scientific investigation has substantial room for improvement. It doesn't seem that science is not good. It's the best that can happen to humans, but we can really make better. There are many possible interventions that may improve the efficiency of research practices and the reproducibility and utility of the evidence. And transparency, openness, and sharing is likely to help, but details on how to can be important, and we need to find out about how these details play out in different settings. A million thanks to all of you for listening, and special thanks to a number of collaborators that have joined forces with me over the years to generate some of the empirical evidence that I shared with you today. Thank you.
You're one of the biggest fans you have, and my lab is definitely, reading your papers. I think it's absolutely true that incentive system is completely wrong. Science, as you showed it, it's really a system, right? And it works based on the incentives that are put into it. I have recently tenured here. In 10 years of my tenure track, nobody ever gave me any credit for reproducibility. Almost when we present data at the conferences, I think we are enemies of majority. When we publish, my lab probably hasn't published study where we didn't include independent validation cohort in the past 5 years. We usually get from reviewers, oh, they should have second independent validation cohort, right? When, 90% of everything that is published or presented doesn't have even the first one, right? So unless we change the incentives, unless we use, for example, these H factors that factor in what kind of methodological, advances we have used or this checklist, right, what we did, whether our work is reproducible, irrespective of whether it's positive or negative, the science will continue the way it is continuing.
I fully sympathize with what you describe, and I think that this is a feeling that many scientists working in very different fields convey to me all the time. But I think that there is progress. I think that there are fields that you have a dominant view that this is important to do. And I think that these fields are likely to be more successful in the long term. And it's not going to happen overnight. I think that if we focus on training younger scientists and hopefully retraining some of the older ones about what really matters and why are we doing this and why it is important to do good science rather than just sloppy science. I think this is like an everyday effort, an everyday struggle. It's not one course that one will take, because I sometimes hear the question, is there one course that I can take? No, this is science in continuity. It's your everyday scientific method and its application, and there's no single course that can replace your experience as a scientist.
Thank you for that talk, and I agree it's going to take a cultural change for this to move forward. Considering we're at NIH and there might be a lot of program staff in the audience, do you have any specific suggestions on the funding side as the major funder of biomedical research?
So I think all of the suggestions that I made clearly apply to NIH, and NIH has tremendous power to facilitate some of these principles. And I think that NIH has moved in that direction on many fronts. Probably not all the movements have been equally evidence-based, but I think that there is people who are receptive to that message, and they do want to change the way that things are done. Clearly, if you have NIH giving priority to these incentive structures, giving priority to openness and sharing, and focusing on good work as opposed to just quick and quote-unquote successful, it can be a tremendous impact.
Yes, there was a high-profile case of Theranos where statisticians called them out and said that this was not going to work, Theranos in terms of the biotech company. And so this was done with incomplete knowledge nor of underlying data from the company. So the question is, what does gut instinct tell you of a reviewer with respect to how something's going to turn out? And it would be interesting if you took the gut instinct of a reviewer and saying, I think this is going to turn out to be profound versus this has turned out to be fake, regardless of the underlying statistical value. Could you comment on that? Because I think that would be really important in terms of biotech IPOs.
So transparency applies just as well in the case of biotech and startups and unicorns. Actually, I was the first to write a critical paper on Theranos in JAMA about a year before John Kairu started publishing his Wall Street Journal investigations. And I sent that to JAMA in 2014. It went through not only review but also legal review, because I was challenging the highest valuation startup in the country that was visited by vice presidents, and all the influential people were on the advisory board, and everybody was so excited, and I was saying they have no evidence to support what they claim. I don't see any paper that they have published. I think their valuation is $9 billion, but it could be just $9 actually. And the paper was published eventually in JAMA. I got a lot of pushback at that time. I heard from their general counsel asking me to recant, and that they were getting FDA approval when they did get their first and single and only FDA approval. All of that is way before the Wall Street Journal. I was told again to recant and write an editorial with their CEO that I was wrong. And I remember Washington Post writing a story like the insanely influential Stanford professor who's asking too much of Theranos. And what a shame, you don't understand basic things. This is how innovation is happening. People now think that Theranos was an exception. It was fraud and everybody's fine otherwise. I don't believe that. A couple of months ago, we published a paper where we looked at every single uniform in the healthcare space. The majority of them look pretty much like Theranos in terms of their transparency and availability of published peer-reviewed science. So I'm not saying it's fraud, but unless we improve transparency and science for these entities, I think that we are running the risk of having deja vu Theranos times 2, times 3, times 4 in the near future.
Thank you.
Hi, thank you very much for that talk. I'm Ian Hutchins, a data scientist here at the NIH, and I spend a lot of time developing and using research assessment metrics for portfolio analysis. And one of the things that I've observed is that there seem to be a lot of cultural and policy barriers to reproducibility efforts. For example, journals will often have in their editorial policies that they won't publish replication studies. I think even PLOS ONE until very recently had that policy in there. And I haven't looked specifically, but one imagines that applications for funding that focus specifically on replicating a previous study don't fare particularly well in the general peer review system. So how do you think that logjam can best be broken up?
So I think we need more training and we need more people to understand what is at stake here. I believe that many journals have changed their stance over time, and many of them are very sympathetic. Many fields that have adopted replication massively, like genetics, would not allow you to publish something unless it's extensively replicated. So it's become a sine qua non. It's the most integral part of the whole world. There's many other journals that are probably lagging behind, and we need training. In a recent study where editors-in-chief of respectable journals were asked to perform an investigation, about a third of them could not even tell that a study was a randomized trial. So the methodologic expertise is often lacking, and we need to just keep pushing that message and educating and training people to be more knowledgeable about how things are working in the way that we do science. I think that the best journals will be the ones that publish the best science eventually, and I think that these will survive. I'm thinking in an evolutionary mode in that regard. I don't want to think that the best journals are going to disappear and we will just get more and more noise.
Thank you very much.
So we've been told we need to exit the auditorium now for another group coming in. However, please, we have people in line to ask questions. Come join us in the NIH library and ask your questions there. Thank you.
Thank you.